Welcome to today's episode where we explore the molecular underpinnings of hormone-driven cancers. Our guest, Professor Luca Magnani from Imperial College London, is a leading expert in the epigenetic mechanisms of breast cancer progression and therapy resistance. His work integrates chromatin profiling, single-cell genomics, and functional genomics to uncover how tumor cells adapt to endocrine therapy. Join us as we discuss how epigenetic plasticity fuels metastasis and what this means for developing next-generation cancer treatments.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
So today I'm joined by Luca Magnani. Thank you, Luca, for joining me today.
Very welcome.
So to ease into the topic, and a question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and in pursuing a career in science?
That's I guess the trickiest of the questions. So I really did not plan much my career. I'm from Rimini, where I am at the moment, and I come from a long-standing tradition of bartenders. So everybody in my family owned a bar. And this is where I started. And then I went to the US to do an internship at the last year of my university. That was veterinary and biotechnology. And when I was there, something just clicked. So I got offered a position for a PhD in animal reproduction. Went back, did my PhD. And then I kind of something again re-clicked in the sense that is a very interesting job. Everything every day is different. And then I just decided to make a career out of it. So I sold the bar and I just stick to science. And then the rest is just random sliding doors.
Maybe can you introduce yourself a bit for those who don't know you already, and what is your current role?
So I'm Luca Magnani. I guess my official role is Professor of Epigenetics at the Institute of Cancer Research in London. And my lab does research primarily in estrogen receptor-positive breast cancer. So we do everything from tumor initiation to tumor evolution and drug resistance, specifically in this disease area.
Mm-hmm. So let's talk about your science in specific. I want to start in 2013. There you demonstrated that genome-wide reprogramming of the chromatin landscape, defined by epigenomic maps for regulatory elements or transcriptional activation and chromatin openness, underlies resistance to endocrine therapy in breast cancer, as you just said. Can you talk about this study and how you got into this?
Yeah, so I have this nature of trying to just annoy people. And at the time I was with my boss, my old boss, Mathieu Lupien. We were at Dartmouth at the time. And the field, was just coming out from TCGA studies. So there was all this massive interest in genetic mutation, copy number changes, and how those were responsible for drug resistance. So everybody was mapping and sequencing. I thought, let's do something different, If I can just be annoying to the field, what would be another mechanism that could introduce drug resistance? And my PhD was in animal science, and I was studying at the time development. So everything in development happens through epigenetic changes, almost everything. So the differentiation from one cell, one germline, one fertilized embryo to all the different tissues is well known to be an epigenetic process. So I thought, why that should not kind of contribute to the development drug resistance. And therefore, we started very easy. We had a couple of cell lines, one that was cultured for month and month with drugs, and eventually become drug resistance. And we just mapped the enhancer landscape of these two cell lines. And what we were able to show is that the enhancer usage in the two cell line was completely different, even though one kind of emanates from the other. So I guess that was one of the first hint in the field that you could reach a completely different cell state, in this case, a drug-resistant cell state, through epigenetic modification. In that particular paper, we didn't do, I guess, anything genomically speaking, but then came afterwards.
Mhm. Yeah, the next thing I wanted to get into, because I found it, if I personally find pioneer transcription factors very interesting and fascinating. And you also had a paper that looked at those. Specifically, you worked on PBX1 and how it regulates the ER alpha transcription response, as you just said, to EGF signaling. So what role does PBX1 play there?
So that was motivated by the fact that the estrogen receptor is an interesting transcription factor. It's a nuclear receptor, so it's a family of 48 nuclear receptor, and differently from other transcription factor, they can be induced. So these are not just simply navigating in the nucleus and occasionally binding to the chromatin. These are induced by ligands, and in this case would be the estrogen. So estrogen signaling, this female hormone, can induce the binding and the transcriptional activation of this transcription factor. The problem is that it needs open chromatin. It cannot just bind everywhere.
Mm-hmm.
So in the field at the time, there was this conundrum that we know there are about 2 million Motif elements in the genome, but there are much, much, much fewer regions in which actually we could see binding. So the question was like, how the estrogen receptor decide where to bind? And this was dictated by this pioneer factor that are transcription factor that can displace nucleosome and open the chromatin in the absence of ligands. And everybody was focusing on FOXA1. That was the most famous. So good friend Jason Carroll was working on that at the time, and Miles Brown before him. So we were just investigating what other potential transcription factor could act as pioneer factor. And we stumbled in this Motif, this PBX1, that is well known in hematopoietic development, but was not really well known in cancer. So by a series of experiments, we could show that a number of estrogen receptor binding was driven and facilitated by PBX1.
But how is then PBX1 regulated in this context?
So that is, again, is something that we then did not pursue to the bitter end. In the sense we could see that was specific probably to the lineage. So estrogen receptor positive cells, even normal cell, are enriched for PBX1, but the estrogen receptor negative lineage, so the myoepithelial cell in the breast, don't express it. So most likely this is a decision, it's just speculation at this time, but there might be something that is dictating the different lineages. So akin to GATA3 and FOXA1. So at some point, there are a lot of people that study how breast develop, but there might be a decision into going into ER positive, ER negative, and most likely there, PBX1 is playing a role. But again, we did not study that in detail.
Yeah, coming back to the epigenetic reprogramming, you demonstrated that drug resistance emerges via drug-specific epigenetic reprogramming. So the thing you alluded to in the cell lines, right? I guess you went into further detail with that then.
Yeah. So what we did, I think, at a certain point in this Nature Medicine paper was to see if there was something similar happening in actual humans. So in that study, we focused on, it was one of the first studies that really focused on epigenetic and tumor from patients. So we put together a collection of about 55 women donors. And these were either primary breast cancer, so newly diagnosed, or were metastatic breast cancer, so breast cancer that come back after treatment. And we use H3K27 acetylation as a marker for active enhancer, and we started to map everything. And that was clearly indicating that there was a different enhancer usage in the primary versus the metastatic setting. And by definition, the metastatic setting is drug resistant. So that was one of the first in patient that through an atlas effort, we could see a differential epigenetic reprogramming happening not only in cell line, but also in humans.
So the readout that you use for epigenetic reprogramming is on enhancers, right? So is it enhancers that get remodeled and hence then lead to differential gene expression?
Yeah, and the reason for it is that 97% of estrogen receptor binding occurs at an answer. So this is something that Jason Carroll, Miles Brown, and Mathieu demonstrated about 20 years ago now, and was unexpected because people really thought that the estrogen receptor would be a classical promoter binding transcription factor. But instead, as I said, 97% occur to 20 kb or more from promoters. And this is then through an induction or an exploitation of loops. So an answer that will loop to the promoter in response to estrogen receptor binding, and this would dictate the transcriptional reprogramming, the transcriptional activation of this gene. And of course, if you use different answer, you will activate different genes.
Yeah. So you did this all by, I guess, using also ChIP-seq. If you do H3K27 acetylation, and maybe this is a little bit nerdy and a little bit into detail, but you also published a paper in Methods in Molecular Biology, providing tips and tricks for ChIP-seq. And this was like, I guess, just before COVID, when I'm not mistaken. But I mean, ChIP is around for 20 years now. So what were those tricks that you published there in this paper?
Because the reason is that at the time, there were hundreds and hundreds of papers focusing on doing ChIP-seq in cell lines. And then therefore you have a limited material, all the cell you want, all the antibody you want. In that particular kind of piece of work, Darren Patton, there was a surgeon that was doing a PhD with me at the time, decided that he needed really to optimize the technology for small biopsy, because we are working, especially if you're looking at metastatic disease, this is not surgical material. We don't have a ton of it. We really have just a small needle that will go into the tumor and get a small kind of like sample. And so he optimized to death the protocol with all the different washes and optimizing the ratio of antibody, beads, and all the rest to make sure that this precious sample always give you decent result, because that's the problem. Before we started, we had a success rate of about 50%, and we ended up with over 90% of success. So now, 9 times out of 10, we could get decent data from a small biopsy.
So are you still using ChIP in your studies nowadays?
So no, we are moving to cut and run. Again, because we could optimize it for smaller cell number, and also because we are moving to ChIP-seq or to cut and run in normal sample, where the frequency of ER-positive cell is much less. So when we look at tumor, these are generally clonal for estrogen receptor-positive cell, because this is giving you a fitness advantage. But if you look in a normal context, there's 1 ER-positive cell every 10. And therefore, ChIP-seq was too noisy for us. And also, this is just dictated by economic costs. So it's much cheaper to do cut and run than doing ChIP-seq.
So is kind of tips and tricks for cut and run also in the works?
It is not going as fast as we could hope. The good news is that we are now in a position in which we could recall all the peaks we call with ChIP-seq with cut and run.
So the recovery is indeed nearly 100%?
Yeah, of the binding site, yes. So now the two technology perform almost equivalent. It's still true that with ChIP-seq, especially for in vitro system, we could always scale it up. With cut and run, that is not like an advantage, because the whole point for us to do this is working with 10,000 cells. But we noticed that when we do ChIP-seq with 100 million cells, of course we get much better signal, because effectively we capture every possible peak. With cut and run, probably we are looking at the strongest, but that in a way is fine, because I think these are probably the most important anyway.
Yeah, that's a good question, right? Are you losing the small outsiders that might have an effect or not? I mean, what is the truth there, right? That's always like the question.
Yeah. So we think, in a way, the estrogen receptor likes to use the chromatin as a parking spot. So out of all the binding site, there's probably a minority that really have transcriptional consequences. And the data in the field seem to suggest that these are probably the strongest one. So all these mid or weak peak are mostly parasitic. Parasitic probably is not a good word, but it's kind of like a parking spot for the estrogen receptor, but not necessarily driving a strong biologist. But again, it would be nice to kind of have a study, some sort of study that would validate this statement. At some point, we're going to get to it.
Okay. So I don't know if you already talked about this, but I asked the question anyway. So you annotated the epigenome of 47 primary and metastatic estrogen receptor-positive breast cancer clinical specimens and inferred phenotypic heterogeneity from the regulatory landscape, a process that you called enhancer mapping. And you identified key regulatory elements commonly shared across those patients. Can you talk about those regulatory elements? Is it like the enhancers, or can you go maybe a little bit more into the details with maybe what genes are regulated?
Yeah, so this was another study that was dictated by me being annoying. At the time, everybody was going super enhancer, and I really hated the term because there's really no science in. Hopefully, people will not get offended with this statement. But like a long time ago, people like Gerd Blobel already defined cluster of enhancers. So we know that enhancers don't work in isolation. They tend to work cooperatively. So all there are, the super enhancer tends to be cluster of important enhancer. But we kind of wanted to demonstrate that. So the rationale of this study was kind of a poor man approach to single cell epigenetics. And the premise was, could it be that the strong peak were simply clonal peak? So all the cells in the sample had that particular histomodification in that spot, while the weak peak were just representing subclonal representation. So only if a proportion of cell had that histone in that location modified. So we did a lot of work in the paper for the first 2 or 3 figures to demonstrate that this was the case. Because, like a nucleosome is a digital signal. You cannot really have a lot of K27. Either you have a lot of nucleosome with K27 or you don't. And then by exploiting this idea, then we started to look at potentially the expansion of some clones. So if we could see, for example, a region, an enhancer that was subclonal in the primary, but then became clonal in the metastatic, this is similar concept of the idea of allele frequency. So if an enhancer becomes important, you would imagine that the cell carrying that enhancer could expand in the primary. And this could tell us something about what is driving resistance. And through this sort of study, we could identify, for example, this transcript that is called SLC93R1. It was previously unknown, but it was clear that the cells that were activating the transcription through enhancer activation were becoming more and more frequent in metastatic setting. And then we did some in vitro work to show that this was linked to the activation of another transcription factor that is called YY1. And together they're activating a program that is specific to metastatic disease. I think the novelty at the time was to show that this was independent from genomic events. So this was not really driven by a new mutation in the metastatic setting, but was driven by the activation of a specific set of enhancers. And now, it's been 7 years we published the study. I think everybody agrees that there are very, very, very little genomic events that explain drug resistance in breast cancer. Beside ER mutation themselves, if you compare primary and metastatic genomically, there's almost no difference. So I think now everybody kind of is on the same wavelength. And this is why epigenetic, I guess, is exploding in the field of drug resistance, because I guess we know that the number of mutations that really explain drug resistance are limited, not only in breast, but in a lot of other tumor. So everybody's just jumping on, what else could really push cells to acquire this drug resistance. And I think we're going to see in the future more and more studies similar to this one, where basically I think people are now using Ataxic as an easier way to get into this. But I'm sure that slowly we're going to see more and more histone modification studies that will look at enhancer activation longitudinally during drug resistance.
So would then, yeah, of course, it's a feedback loop, right? You treat the cancer with a drug, and then it gets resistant to the drug because of the drug treatment, right? I mean, it was not there beforehand.
There's an adaptation, right? So this is just an evolutionary question. You stress the cell, and the cell need to reroute their evolutionary trajectory from what they were doing before, that was simply proliferate as fast as you can, to, well, we need to survive, and then we need again to select for something that is proliferative. I guess that the conceptual difference is that people were hoping for mutational biomarkers. So the idea was like, you give a drug, you get a specific mutation, and then we design a drug for the new mutation, and everybody's happy and things are straightforward. And we know that that's not the case in a lot of cases, right? For breast cancer, I guess we know that 30% to 40 come up. 30% of the cases that are long-term treated with endocrine therapy eventually develop an ER mutation, but that is not the sole explanation for drug resistance, because even drugs that target, yes, the mutant estrogen receptor don't cure patient anymore. So there's a constant epigenetic adaptation to whatever we throw to these cells that needs to be understood, and we need to be smart in circumventing this, because the idea is really eradication. Like the next frontier for this tumor is not simply prolonging the response and therefore prolonging the survival. We really want to get to a curative state for metastatic breast cancer, and we're not yet, unfortunately.
So you then took the single cell approaches further and compared early and late phase of the endocrine therapy, and what kind of differences you have there in transcriptional plasticity. Did you then do a true single cell experiment?
Yeah, so we went from being alternative music to mainstream. Everybody was. This was 2019, so single cell was there. We thought, like, you know what? If you cannot beat them, you join them. At the time, there were not really straightforward way to do single cell epigenomic. But single cell RNA sequencing was much more robust. So in a way, we kind of went for a confirmation. We did 3 studies in a series. The first one was just a simple single cell RNA sequencing, where we showed that there is phenotypic plasticity, and there is, even in untreated sample, there is this pocket of very pessimistic cells that already have the feature of a drug persister. So the drug is not there, but they think that there is drugs. And we call them the Cassandra cells. So they are just simply complaining about everything, even if everything is fine. But then the problem came because the number of pre-existent cells was not enough to explain why we saw so many persister after therapy. So it became a question of how many are induced and how many are selected in absence of mutation. And this is where we did a couple of studies we published last year in Cancer Discovery, where we basically look at leveraging lineage tracing and single cell in the same experiment. So now we could kind of look at the last name of a cell and see how that family of cell evolved, transcriptionally speaking. And that was very informative, because we saw that the vast majority of persister are induced by the treatment. And now I guess this is not surprising anymore. But the idea, is that there's very little that is selected straight. So these pessimistic cells represent a pocket of persister, but they are a vast minority. Most of the cells really make this decision into, should I die or should I go dormant and persist? And vast majority do that. So they really kind of decide to go dormant for a long time. waiting for a better future.
Yeah, so you were looking for the last common shared ancestor, basically.
Yeah, and we kind of like, the interesting thing is like we showed that the last common ancestor of all the persistor cells is stochastically defined. So there's nothing you can really say in an in vitro system that could predict, okay, this cell will become persistor and this cell will die. Every time you do an experiment is kind of a Marvel comic. You have multiple universe. And this became a problem because all the concept of replicate went to the bin. So we started to do CRISPR screen and realized, we could not analyze them because every experiment was its own story. If we took two lineages and we look at the same lineages in 10 replicate, in some replicate, this lineage managed to go into dormancy. In another replicate, the same lineage will decide to just die. And then it's complicated because you're looking for, you're tracing, you're doing lineage tracing, and things are just not reproducible. So it became, this is where the lab went a bit mathematical crazy because we tried to model these things and it became more and more complicated. But I think in the process we learned so much. And then, of course, now we're trying to do everything in vivo because I'm a fan of sci-fi in vitro work, but I think at some point we really need to make sure that this is what's happening in women and in patients.
What you also looked at, and you just mentioned that, so cells go dormant, and then they sit there, and maybe after a couple of years or even a decade, they awaken again, and then they grow again. So you also looked at this process, right? At what happens during the awakening process.
And it was really intriguing because even in vitro, if we start with 10 flasks and we treat these 10 flasks with therapy, they all go dormant, but then the waking up was completely asynchronous. So we had one flask that woke up after, say, 3 months. And then we had the second flask that woke up after 9 months. And our record holder was a flask that after 18 months was still dormant. So it became impossible to really predict how will they wake up. And this, again, I think it's very important. Again, we don't have enough data to say that this is exactly what happened in vivo, but this would predict that in women, if you were to clone the same tumor in the same woman and just let it go for 10 alternative universe, you will get completely different waking up dynamics. And this is why we are pushing so hard into eradication. Because if we're just waiting, for a biomarker that will tell us, okay, now the tumor is ready to wake up, let's do something, it's going to be tricky because every time is different. But yeah, so the funny thing is that the postdoc were not really happy. This is a study that we could do only because COVID hit. So it's a positive effect of COVID because everybody abandoned the department and we were given all the incubators.
Yeah, I can imagine that if you have to split cells for 18 months, it's...
Yeah. So, and then we did this in hyperflask. So these were billions of cells. These were this 20 liters flask because we wanted to make sure that they had, we didn't want a split cell. We just wanted to wait. So interesting times.
Were they still growing, or what was the state when they are dormant? Is it like a senescent-like state, a quiescent-like state?
So it's a good question. So it's depending what assay do you use. If you look at it genetically, they have some feature of senescence. For example, they accumulate a lot of heterochromatin, including the very mysterious H4K20 trimethylation. But then if you look transcriptionally, there is some sort of overlap with senescence. But then if you look with beta-gal staining, for example, a classical marker, you don't see any of it. So we always get this sort of question at conferences. And I think, it's just a question of how do you define senescence. Senescence is generally defined in normal cells. So if you look at fibroblasts, they go senescent. If you look at endothelial cells, they can go senescent. And I always kind of think like, can cancer cell go through senescence? So what we've done to really try to get a feeling for this was treat them with every possible senolytic that was available. And senolytics do not really kill these dormant cells. So I guess functionally, they are not really senescent, but they share some of the program of senescence.
Mm-hmm. Yeah, interesting, because if you are truly senescent, then you probably won't also be awakened. So, yeah.
Yeah, that's the thing. But there is evidence that I think a very old study from Judy Campisi showed that if you forget your senescent cell somewhere in the incubator, at some point they can survive and reawaken even fibroblasts, but it's it's ultra rare. So we've tried with UVEC to get them to leave them there for a long time and see if they woke up. And this is all unpublished. We're gonna hopefully publish this next year, but we could never see this. So I think maybe the barrier is much higher
Mhm.
for normal cells.
So imagine you are due to submitting a grant proposal tomorrow, which you probably are. What would you have written into this?
So what I'm writing right now is really solving the next bottleneck in estrogen receptor positive biology, that is the lack of animal models. It's surprising to say, but this is the most common disease in women, right? This is the most common cancer in women. It's still the number 2 killer in women. We have done so much. I mean, it's incredible the amount of drugs we have, the stuff we know, and everything is coming from a panel of cell line. Mouse model are really refractory to go hormone dependent breast cancer. They are fantastic tools if you want to model triple negative breast cancer or HER2 positive, so the other two subtype. But the most common subtype, really difficult. So we don't have animal model to do this. So we wrote a grant and we are pursuing this despite anything, despite all the rest, because there is a rat model that used to be used in the 70s and 80s. So again, if you think about a musical career, we went alternative music, then we went mainstream, and now we're going to go revival and back to the 70s. And these particular rats, they develop ER positive breast cancer. They need the line, this strain has been frozen a long time ago, so we are resurrecting it. So all the effort in the lab in terms of breast cancer is going to go towards this repeating some of these studies. So really repeating all this lineage tracing, single cell epigenetics, epigenomic, but in an immune competent syngeneic rat. If we can achieve that, I think we are going to be in an excellent position to really move forward. And the second thing we are doing, the second grant we are really dedicating, is understanding tumor initiation, because I think that is another massive gap in the field. And for that, we are developing clinical trials in healthy women to really understand what the estrogen receptor does in the normal context. But again, surprisingly enough, we don't know. We know a lot in cancer, but we don't know nearly enough in normal cells.
But I guess if you initiate this clinical trial, this has like a long time horizon, right? I mean, this will.
So relatively, the normal one now is going to be relatively short term, because basically what we're going to do is to look into the normal menstrual cycle and use the fluctuation in a normal menstrual cycle as a readout for activation of the estrogen receptor. Because as I said, this really responds to ligand. So if we can kind of compare within the same woman what happened when the ligand is low and when the ligand is high, hopefully we will get a feeling of what it does in this normal cell. And then hopefully by comparing it to a cancer, we could start to understand that genetically speaking, why does it become an oncogene? Why the estrogen receptor promotes proliferation when it probably doesn't in a normal setting? And then we will build up, of course, all our epigenetic love affair. So we're going to start to look at epigenetic usage in normal versus cancer.
So for the last 35 minutes, we've been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I always like to say something to these you hear PhD student and postdoc that are, of course, the future, right? So I'm already old school. I think that I'm always very positive that the good stuff will come from the next generation, but the next generation is always scared into going through their career. They always think that they really need to get the paper right, get the grant right, and really follow a predetermined trajectory. And I'm always reinforcing this idea that don't worry. Like if you really like science, things always work out in a way or the other. And there should be less stress in the field. People should approach science with more enthusiasm and a bit more open-minded rather than really prefixing all these predetermined goals. Just follow the science. Things will happen, and generally are always nice.
Yeah, you just have to take the opportunity when it presents itself. But there is also so much outside pressure, when you look at the political environment.
Ignore it. You cannot. That is something you cannot do anything about. So the only thing you can do is enjoy your work. And then if tomorrow we have a job, that's fine. If tomorrow we don't have a job, go back to bartending. There's always a solution.
That's very true. So my last question always is, what is your personal definition of epigenetics?
I don't know. I'll give an annoying answer for the field. It's not attack. So I believe is the fundamental thing that we are missing, and I'm not the right person to solve that, but it has to be an inheritance form of information. So is epigenetic where we really understand how a cell can transmit that bit of information to the next cell. So histone modification, DNA methylation, fantastic. Open chromatin, I don't know. There might be a correlation in there for sure, but I don't know what that would be. So I really hope that in the field of true epigenetics, that is not what I do. I really appreciate people that really are hardcore in epigenetics. We really need to solve this because otherwise it becomes a correlative field, and that's not good. We need to get to the mechanism of epigenetics.
So thank you, Luca, for your time and for being on the show.
Thank you very much for hosting me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.